who, we're also recording this meeting. Dr. Holmberg is a research scientist here at Downeast Institute, specializing in marine carbonate chemistry. So he's going to give a lot of information about ocean acidification. He has a PhD from UMass Boston, and he joined DEI in 2019, where right off the bat, he built our state-of-the-art ocean acidification laboratory that allows us to do experiments about ocean acidification as well as increased temperatures and see what sort of effects are happening to different marine organisms. He began lab experiments once he got the laboratory set up, and he's been going full steam ahead, doing a bunch of different work in the lab and also in the field to try to understand what's happening in the wild with especially softshell clam populations. So with that, we'll hand it over to Rob.
Hi, everyone. Thank you so much for being here. I just want to start by saying that Ray and I are about a full month into an ongoing exposure trial. So please bear with us as we're operating with limited brain power.
So you have to share this presentation.
We're just pulling the presentation up.
It's on the computer. Oh, yeah.
All right, here we go. So first I'd like to give a brief overview of ocean acidification for those of you who are not already familiar. As atmospheric CO2 rises due to human activity, a lot of it is dissolving in the surface ocean, causing ocean CO2 to rise in parallel. This causes surface ocean pH to drop in a process called ocean acidification, or OA. Pulling the clock back 300 million years, it's still fairly high, but it's expected to drop to 7.8 by the year 2100. This change may happen faster than ever before in recorded geological history, and it's likely to outpace the rate of adaptation of many marine species. This is why scientists are so concerned about OA. So these are the simplified equilibria that govern the carbonate chemistry system in the ocean. When you add CO2 to the system, the equilibria shift in such a way that one, seawater pH decreases, hence acidification, and two, more of the carbon in seawater takes the form of bicarbonate rather than carbonate ions. Both effects have implications for the physiology of marine organisms. Another way to think about this is by plotting seawater carbon species abundances as a function of pH. As you can see, as seawater becomes more acidic, carbonate decreases and bicarbonate increases. In other words, the more bicarb you have, the less calcium carbonate you have. And that could be calcium carbonate dissolved in water or in sediments on the seafloor or in the shells of marine calcifiers. We quantify the abundance of calcium carbonate as the saturation state of aragonite, which is one form of calcium carbonate. Calcifiers like it when the saturation state is high, but OA is pushing it lower. And coastal systems like the Gulf of Maine are under pressure from OA like the rest of the ocean, but they have certain characteristics that make them even more vulnerable. For example, rivers like the Penobscot and Kennebec input large volumes of fresh water, locally diluting aragonite saturation, and riverine input is increasing with precipitation due to climate change. Also, coastal eutrophication from waste effluent causes algae blooms, which then die and decompose, causing acidification. And in the Gulf of Maine, there's an inflow of cold, low aragonite seawater at surface via the Labrador Current, which itself is freshening due to climate change-induced Arctic ice melt. All these factors and more are contributing to rapid acidification in the Gulf of Maine, and all regions therein will reach the 1.5 aragonite saturation threshold by the year 2050 under a business as usual CO2 emission scenario. The 1.5 threshold is significant because it's the threshold that was identified by researchers as one of disruption to calcification and development in many marine calcifiers. These are a handful of species that are not only ecologically important, but also economically important in Maine, which are either fished commercially and or produced with aquaculture. Collectively, these fisheries are valued at many millions of dollars, and it's for this reason, along with the proportion of harvest and the number of fisheries jobs, that Eastern Maine was ranked among the top 20% of coastal regions in the US for high economic sensitivity to OA. Suffice to say, Maine may have a lot to lose from ocean acidification. OA is sometimes referred to as the other CO2 problem. The original CO2 problem, of course, being global warming due to the greenhouse effect in the atmosphere, which is also warming the surface ocean. The Gulf of Maine warmed faster than 99% of the global ocean between 2004 and 2013, attributed largely to changes in ocean circulation, and in particular, a northward shift in the Gulf Stream that pushed warm seawater through the Northeast Channel. Still, about 1/3 of the recent warming trend is attributed to anthropogenic warming, and greenhouse gas emissions are expected to warm the Gulf of Maine up to 3.5 degrees Celsius beyond recent temperatures by the year 2100. And while ocean warming dampens the severity of OA somewhat, OA and warming have interactive and negative effects on many marine organisms. Understanding the impacts of OA and warming on different species and life stages is critically important for management and mitigation. This mess of wires and tubes is the OA system that I built down the hall here at DEI. It allows me to manipulate seawater pH and temp in each of 30 experimental aquaria with true statistical independence and calibrate my probes directly to synthetic seawater buffers for monitoring pH on the total hydrogen ion concentration scale, which is the scale that marine chemists use to measure pH. We're using it to run a series of exposure trials featuring the larvae of various Maine bivalves and crustaceans. So last summer, I used it to complete an experiment studying OA and warming impacts on the commercially important soft-shell clam, Maya arenaria. I used a full factorial experimental design, including 3 seawater pH treatments, 2 seawater temp treatments, and 3 replicate tanks per treatment. I reared Maya for 2 weeks from hatch to settlement and then quantified differential mortality and shell morphometrics. I wish I had more to show at this stage, but I'm very early into the analysis, so here's a sneak preview. In a random intercept multi-level model in which response data is nested within the tank factor, pH, but not temperature, influenced Maya shell length. There was no interaction between pH and temp. This bar plot shows combined means by treatment with 95% confidence intervals calculated from pooled standard deviation. The CLD shows pairwise differences between each combination of treatment levels. Briefly, clams grew about 7% smaller in the pH 7.2 treatments relative to the 7.8 controls, regardless of temp. Following a planktonic larval phase, Maya settle in the mud where they remain for the rest of their life cycle. Currently, Maya is under intense pressure from predation by the invasive European green crab, which is increasing due to ocean warming and decimating population numbers. Moving forward, they'll face the double whammy of predation and OA pressure as well. Sediment porewater on tidal mudflats is already acidified relative to the water column due to microbial breakdown of organic matter, but scientists are concerned that OA will push it below its natural minima. And for all of the talk of the importance of diel and seasonal pH cycles in the coastal ocean environment, I was surprised to learn that there are no available long-term data sets on carbonate chemistry in tidal mudflats. That means we don't really have a good understanding of how the pH and aragonite saturation change through the year, which means we're not really in a good position to predict how OA will impact it moving forward. We're aiming to rectify that with the generous support of the Broad Reach Fund's main shellfish restoration and resilience project. We're collecting porewater pH and alkalinity samples from 2 of DEI's Clam Recruitment Monitoring Network sites in Beals on a monthly basis through the entire year for a complete picture of the carbonate chemistry cycle. And we're doing this in collaboration with Butch Pinkham, the local shellfish warden, who is off screen collecting mud samples. Having said that, scientists are already exploring mitigation strategies for counteracting sediment porewater acidification to benefit Maya and other mud inhabitants. One strategy involves spreading crushed oyster shell, or shell hash, on mudflats to buffer acidification. However, of the 9 existing studies that tested this method, only about half of them observed any effect of the shell hash. This could be in part because every study used different sizes and densities of shell, resulting in different outcomes. DEI is working on a shell hash project in collaboration with the Maine Coastal Program, DMR, the Casco Bay Estuary Partnership, and MIT Sea Grant to try and crack the code and determine which combinations of shell size and density are most effective for buffering sediment porewater acidification. We started with a lab experiment in which I collected 55 mud cores, set them up in a flow-through tank programmed to simulate the tide, and spread 3 sizes and 3 densities of shell hash in a factorial experiment, leaving them to sit for 2 weeks before analyzing the carbonate chemistry. All of the shell was recycled from seafood restaurants. Interestingly, only shell size and not shell density impacted porewater carbonate chemistry, and only the smallest shell size boosted pH, alkalinity, and aragonite saturation. And while the results of the lab experiment are compelling, what we're really trying to do is follow through and create a solution that has tangible benefits for Maya and for communities. To that end, back in April, we set up a large-scale field experiment on a tidal mudflat behind Hannaford in South Portland, featuring 3 shell size treatments, 3 shell density treatments, and 2 predator deterrent netting treatments. We set up the experiment just prior to Maya settlement and sampled porewater carbonate chemistry immediately before and after shell deployment, as well as a month following shell deployment. We'll sample carbonate chemistry another 2 times before quantifying Maya abundance at the end of its growing season in November. And by then we'll know whether the effect of shell hash buffering lasts for a full growing season and whether it's worth the trouble for communities to implement it. So that's it for my presentation, and I'll take any questions.
Does anybody have questions? Oh, see Teresa.
Okay.
Parker. Oh, that's a clap, sorry.
Nessie?
Can you give an example of what can happen to a marine calcifer under OA?
Yeah, sure thing. So both pH and the saturation state of aragonite influence calcifiers in such a way that it makes them difficult for them to calcify their shells and also maintain homeostasis. So a really extreme example is one that was detailed in the seminal paper by Mark Green called Death by Dissolution, in which Maya settling into the mudflat literally dissolved from the acidified conditions. And sometimes you just see like little bits of pitting in the shells, which might weaken that animal's defenses if it's a bivalve and they need their shells to defend from predation. And in other times, the mortality is quicker and more direct.
Mark Whiting has a question in the chat.
Why shell hash and not agricultural line?
So our project is about recycling oyster shells from the seafood industry. And it feels more holistic that way by taking this calcium carbonate that was removed from the ocean and then returning it to the ocean in such a way that benefits mudflat inhabitants.
Bassem Alam, sorry, I might be butchering your name.
Hi, Rob. Great presentation. I wonder why the shell size matters. I know that the shell composition can change over the age of the oyster. It was oyster shell, correct? So why you think you have differences?
We think it has to do with dissolution kinetics. And the simple answer is smaller bits of shell dissolve more efficiently and become incorporated into the pore and the sediment.
And Parker?
Thanks, Rob. Thanks all. As a follow-up question to Bassam, really exciting, Rob, as you mentioned that you're starting to crack that code around what size shell is most influencing carbonate chemistry, and yet in the constraints of implementing this kind of approach towards OA, we know that there's enormous costs associated with crushing shell as opposed to just bringing the raw material back into the environment. And yet we also know that when whole shells are put into a marine environment, over time, they'll naturally crush into a smaller aggregate and dissolve into a smaller aggregate. So I was curious, with your experimental design hat on, is there a way to understand how long managers could expect for whole shells to be deposited into an ecosystem before they're actually ready to contribute that ideal buffering capacity?
Sure. So that's kind of outside the scope of this particular experiment, but it's something I'd be interested in learning more about, certainly. But as I said, we're finding that the smaller bits of shell work best and the larger bits of shell don't work at all. So we don't have any evidence to suggest that laying down whole oyster shells would do the trick. You know, at least with any kind of immediacy.
I've just got a comment on that one. So that's a great comment, Parker, about adding whole shell. The problem is it is probably going to take years for that to dissolve. And in the meantime, that shell is acting not only perhaps as a buffer, but it's also adding habitat to that sediment surface. And that habitat can be colonized by everything from shrimp and small fish at high tide to crabs and other smaller predators. So I think that there would be a cost associated with that in terms of mortality to shellfish as a result of the habitat that oyster shell, whole oyster shell, would bring to the environment.
Yep, and I just want to comment on the cost of crushing shell. So initially we looked at very large, very expensive machines like hammer mills that could crush the shell in industrial quantities. But you know, we thought about how that might be implemented by communities and decided that that was not only outside the scope of our research budget, but it would also be outside the scope of the budgets of communities. And so we struggled with this problem for a long time, but luckily DEI has a world-class facilities crew composed of 2 Jeffs, and they took a $700 glass crusher and modified it to turn it into an oyster shell crusher. And so we're actually achieving this really cheaply.
George in the chat asks, would agricultural lime be chemically interchangeable with shell material?
Sorry, could you repeat that?
Would agricultural lime be chemically interchangeable with shell material?
I would expect it to be, yeah.
Any other questions?
Well, thank you, Rob, for that presentation. Now you know how to get rid of.
Thanks again, everyone, and we'll pass the floor to Ray, who's going to talk about the Atlantic Surf Plan.
Yep. So next we have Ray Czaja, who has been working at the Downeast Institute this summer on experiments dealing with Atlantic surf clams. And he actually also was, this is not his first time at DEI because he also used to work with Dr. Phil Yund a few years ago doing some other work. And he's a PhD candidate at Stony Brook University in New York.
Hi, everyone. Thank you, Sarah, for that introduction. As Sarah pointed out, I'm a PhD student from the School of Marine and Atmospheric Sciences at Stony Brook University. More specifically, I'm part of the Marine Animal Disease Lab. We specialize in all sorts of shellfish biology. And so I'm here primarily funded by a New York Sea Grant project where our goal is to look at the effects of climate change on surf clams, specifically the New York surf clam fishery. We have a few questions that we want to answer. One of those questions is, what are the impacts of OA and temperature increases on surf clam larvae? And luckily, Rob gave a very nice introduction on OA, so I don't have to struggle through all that chemistry. And instead, I get to talk about what I'm more specialized in, but frankly interested in, and that's fisheries ecology, fisheries oceanography, biological oceanography, those sorts of questions. And so I am first going to begin with a brief introduction on the bivalve reproductive cycle and what happens to larvae when they're in the water column. This may be old news for many of you, but in case you're not familiar with it, why not go through it? So bivalves are broadcast formulas, right? They release sperm, they release eggs, they cross their fingers, and they hope that fertilization happens. If fertilization happens, they get an embryo. That embryo turns into a trophophore, trophophore turns into a veliger, veliger turns into a full-grown larva, and eventually that larva will settle. Before it settles, it's hanging out in the water column. It's doing a bunch of things, right? It's growing, it's eating, it's respiring, some may die. But the thing that I'm mainly interested in is the fact that it's dispersing, it's moving. It's fertilized at one location, it's moving along a path that reaches another location, then it settles to the seafloor. I'm interested in what those paths are and what causes those paths to be what they are. But so yes, eventually they settle, and then after they settle and they grow a bit, they recruit. Now, recruitment is a very broad idea. People define recruitment different ways. The main definition for recruitment is the process by which an individual enters the population. It's pretty broad, right? People can fine-tune recruitment for their question of interest or their context. And so for the research here, I'm quantifying or I'm calling recruitment the starting point of fertilization and an end point of when the surf clam grows to 3 years old. And why 3 years old? You can say, oh, well, it should be 1 year old because surf clams can actually reproduce at or before age 1. The reason I'm saying recruitment ends at age 3 is because before age 3, they are too small to be consistently caught by both fishing gear and serving gear. And so a lot of the data we're working with deals with, the youngest clams being 3 years old. And so why do we care about recruitment? Well, you can't have sustainable population if you don't have individuals entering the population, if you don't have recruitment. And you can't have a sustainable fishery if you don't have a self-maintaining population. So I'm going to switch gears a bit and talk about some relevant information about the surf clam fishery. So the Atlantic surf clam is Bacillus solidissima. It's a relatively large bivalve, a few inches long. And here in the Northeast US, it supports a near $30 million fishery. It's primarily used for fried clam fruits and chowder, among other things. And its traditional distribution is between the Gulf of St. Lawrence and Cape Hatteras. And its traditional habitat is in shelf waters between 10 and 50 meters deep. I say traditional because around here, it can actually be found in the lower intertidal zone. But I think that's mainly a exclusive Gulf of Maine phenomenon. And also, it's primarily a captured species, but again, Maine kind of a different beast. And around here, it's actually people's VEI can sell seed to some farmers, and the farmers can raise them as juveniles. And I've been told that the juveniles, that yay big, are quite tasty when served over pasta, which is obviously, you can't get a clam that's this big, the normal capture methods in other parts of the fishery. So now I want to transition to geography for a bit. Again, this may be a review for some of you, but in case we're not familiar with some of these regions. Like I said, the Atlantic surf clam distribute from Cape Hatteras to Nova Scotia. That being said, its primary capture fishery is in the mid-Atlantic bight, that blue region on the map here. Again, it is found in the Gulf of Maine. And I'm particularly concerned with within the mid-Atlantic bight, the New York bight, this stretch or this area of water here. And if I wanted to be even more specific, I would say I'm concerned with the waters of the south shore of Long Island. So that would be right over here. A lot of the data that I'm going to be referencing is coming from this area or off the coast of New Jersey or even down here in the Delmarva. So what do we know about population trends with surf clams and the status of the fishery? Well, we know it's not good. Biomass and along with growth and health issues have been causing issues for surf clams. You can see here over this 25-year stretch, biomass in the total mid-Atlantic bite, MAB, whoops. Can I not go back? There we go. Has been decreasing for nearly 20 years. So it's not a good thing, obviously. And recruitment study that I'm working on that's not yet published, this is kind of a sneak peek here, shows that in the New York Bight, recruitment has been decreasing-ish since 1987. So this red line, this is predicted recruitment from the models. And yes, there's some spikes, and yes, this observed spike is over here. But over this near 25 years span, you see a slightly negative slope. So again, decreasing recruitment in the New York Bight, at least in western Long Island waters. And we also know that biomass is decreasing inshore. So if we take a look in some slope waters, or rather shelf waters in the Delmarva region, so this I believe is Chesapeake Bay right here. Oops. So back in 1986, that green inshore, that shows that there were a fair amount of surf clams in those inshore waters. But fast forward to 2008 in plot B, you can see that surf clams are virtually absent from those inshore waters in the Delmarva region. And we also know that overfishing is taking place. So if it's not overfishing, what's causing all of these, negative responses in the surf clam fishery? The best guess is climate change, right? And the usual suspects are increasing temperature and ocean acidification. And we actually know quite a few things about how surf clams respond to increasing temperature and OA stress. So we know that adults don't like it warm. You look at this figure here, we have scope for growth plotted on the y-axis and energy. Scope for growth is an experimental way to estimate how much energy an animal has available for growth. And so we can see the blue box shows that at 19 degrees Celsius, adult surf clams have a nice positive scope for growth. That means they are taking in more energy than they are using for respiration, maintenance costs, excretion activities. However, when we bump up temperature to 23 degrees Celsius, we can see scope for growth becomes significantly negative. So essentially what that means is that increase of 4 degrees Celsius causes the surf clams to use more energy than they are taking in. And backing up to my recruitment model that I'm fairly proud of. I talked about this these predicted recruitment trends, right? Well, what are they predicted from? On our models, we threw about a dozen predictor variables in there. And for Western waters, the only two predictors that stuck as significant were summer and fall sea surface temperature. And those relationships were not positive, they were negative. So what that means is during a year when we have a warm fall or a warm summer, we have poor recruitment. So all these things point to, again, temperature stress being an issue for surf clams. And we also know quite a few things, as of very recently, about how surf clams respond to ocean acidification. So a recent study by Emilian, who may be in the audience today, found that at a pH of 7.28 or PCO2 concentration of a little over 2,000, surf clams, adult surf clams, have a significantly lower scope for growth than they do at two lower PCO2 concentrations, two higher pHs. And from a study that was published literally a month ago, we know a bit about how surf clam larvae are responding to OA. So we know, or this figure shows that at a moderate drop in pH to 7.63, the larvae actually increase their growth rate. But when we drop the pH down to 7.46, the larvae then decrease their growth rate. So we know quite a bit already, but there's also obviously tons of things that we don't know. I keep talking about my recruitment model, my relationship there, but we don't know what the mechanism behind that is. We don't know why there's a negative relationship between fall and summer sea surface temperature and recruitment. We don't know if OA interacts with warming effects. And we also don't know if OA and warming affect larvae behavior. And the reason I believe behavior effects could be important is because behavior effects may scale up to changes in dispersal patterns, dispersal distances, and sediment locations. I'm going to talk more about how those things are directly linked in a few moments. But again, recruitment decreasing. Temperature plays a role, but what is that role? And are other factors such as OA at play? And if they are at play, again, what are the mechanisms behind these effects? Why are these stressors leading to reduced growth or increased mortality and decreased recruitment? So there's a few different reasons why this could be happening. If growth rates are too slow, larvae are hanging out in the water column longer. Maybe they're having different interactions with hydrodynamic forces. Maybe they're more susceptible to predators if they're taking longer to grow and if they're smaller. Maybe simply that they're just dying. They're falling out of the water column. They're using too much energy. They're respiring too much, and they're not taking in enough energy from the phytoplankton and the other food sources around them. And here's my bread and butter. I believe that these climate change stressors are going to be inducing behavioral changes. And what do I mean by behavior? I mean swimming. And yes, bivalve larvae actually swim, even though they're microscopic. They can swim up to 1 millimeter a second for some species and under certain scenarios. And when they swim, they are primarily swimming up. They're not swimming with the current or against the current or down. They're primarily swimming up. And the reason this is important is because if they're swimming up, that means they're changing their vertical position in the water column. And if they're changing their vertical position in the water column, that could mean that they're either above the thermocline in the surface mixed layer or below the thermocline. And if they're swimming up and they're in that surface mixed layer, they are in a layer of water that is essentially at the mercy of the winds, and it's experiencing more wind-driven flow. And so the larvae that are in that surface mixed layer and are experiencing more wind-driven flow than larvae that are maybe below the surface mixed layer and experiencing less, if not absent, wind-driven flow, they're going to be on all different trajectories, all different speeds. They're going to be winding up in all different places. And so I believe that behavior changes will eventually scale up to differences in dispersal patterns via that mechanism that I just described. So again, behavioral changes can be maybe recruitment isn't decreasing overall. Maybe recruitment is just decreasing in the spots that we're looking, but it's increasing in other spots, such as offshore. And again, these are questions that could potentially be the answer to some of the plans that we have. Or maybe it's combo package. Maybe temperature and OA and stress are affecting growth, or affecting mortality, affecting feeding, affecting the interactions with the hydrodynamic environment. Maybe during 1 year, when it's particularly warm and there's a particularly low pH, the larvae aren't growing that well. They're lower in the water column and they're settling at this location. But then during another year, when it's cooler, a higher pH, they're less stressed out, they're swimming more, they're higher up in the water column, they're experiencing more wind-driven flow, they then settle over here. And so let's say raise crazy theory of behavioral changes leading to differences in water column position, leading to differences in hydrodynamic forces that they experience, leading to differences in dispersal patterns, leading to differences in sediment location. Let's say all those links actually exist and all these things are happening. Well, what's next? If let's say we can actually predict where the surf clam larvae are settling, what good does that do us? And so the idea is if we can predict where they're settling, we can potentially increase fishing yields by targeting the more productive areas. Or rather, the more responsible way to do this would be if we can predict where they settle, again, which is a big if, you stop fishing in that spot for a year. You let those post sets, grow, or maybe even more than a year until they are a couple years old, they've reproduced enough times, and they've recruited to the fishery. And then a couple of years later, the fishermen come in and they can take all what they want. And so, if we can achieve this kind of a nice balance of allowing the population to maintain itself, but also allowing the fishermen to be more efficient by targeting specific areas and wasting less resources. And so the next question is, okay, Ray, I believe you, but how the heck are you going to make these predictions? And so in the physical oceanography world, if you're interested in what's happening to a particular particle in the water column, in our case, a larva, you can take two approaches. You can take what's called a Eulerian approach. The idea with the Eulerian approach is that you pick a point in our case, the ocean, and you measure what's happening to that point through time. So, in this case, it would be like putting like a plankton net at one particular spot for X amount of time and catching plankton at that one spot. But if we want to try to take that, application and put it in our context, it's like searching for a needle in a haystack. When the needle moves, the haystack is ginormous, and the amount of money it takes to use the equipment to catch that needle is incredible. So these types of approaches for larval transport, larval dispersal, aren't exactly practical because of the sporadic and patchy nature of larvae in the ocean, particularly in shelf waters too. So instead, scientists typically use what are called Lagrangian approaches. And with Lagrangian approaches, instead of picking one spot in space and measuring what's happening to that spot through time, you track your particle of interest. You move with the particle instead of staying in one spot. And so some of the more traditional ways of using Lagrangian approaches to measure particle transport in the ocean, particularly larval transport, include genetic techniques, artificial tags, and natural biogeochemical tags. The idea behind these three approaches is that you get some recently settled larvae at location A, right? And then you get a few other locations, and you get some surf clams from those locations. And then you measure the genetic signature of those surf clams, or you measure what the biogeochemical signature is of the shell. And then you match those signatures of your post-set larvae in location A to all the other locations, and you see which is the best match. And whichever is the best match, you can then say, okay, those two areas, those two subpopulations, those two metapopulations are more connected than others. And assuming you have some idea of what the physics are, you can say, okay, well, these animals are upstream, these animals are downstream. So these animals function as a source population, these animals function as a sink population. And these approaches I just mentioned, are awesome for those types of questions, for metapopulation questions, for connectivity questions, for source sink dynamics. But they're not ideal for making predictions. And the reason for that is, with the biogeochemical approach, with the genetic approach, you're limited to the specimens that you have on hand. You can only make conclusions about the areas from which you have specimens. You can't make conclusions about other areas that you haven't sampled, other areas where you couldn't get clams from, or even other spaces or other time points. And this is where biophysical models come in. Because with biophysical models, you can at least attempt to predict certain scenarios in time and space that you can't get specimens for. And so the idea behind a biophysical model is kind of just like it sounds. It's a model, it's a giant equation, you put into a computer with a lot of computing power. You put in some physics variables, you put in some biological variables, and then that equation spits out, your dispersal pattern metric of interest, the dispersal path, the dispersal distance, the settlement location, the percent settlement success. All these things can be quantified with biophysical models. And so by the physics, there's a lot of inputs that go into the physical component of the model. Some of the big ones are obviously currents and flow, buoyancy-driven flow, density-driven flow, wind-driven flow for that surface mix layer I was talking about, cross-shore transport, long-shore transport, tides in certain areas have huge roles, particularly around here. And temperature is a big one too, because temperature affects the depth at which that thermocline occurs, and therefore how wide that surface mix layer is. And then there's the biology side of these biophysical models. You put in growth, you put in size. You also put in time in the water column, or you can say this is also the time to settlement, or as the biophysical modelers call it, PLD, pelagic larval duration. It's the duration that the larvae are spending in the pelagic zone. Essentially, it's the amount of days it takes from fertilization to settlement. And there's other metrics you put in there too. Sinking speed, swimming speed. And again, all these things can give you your dispersal pattern metrics of interest. And so a recent study, actually, I guess it's not so recent anymore, from a group of scientists down in Rutgers they kind of laid a really nice foundation for what we know about surf clam larval dispersal already. So we know what the large scale geographic patterns are throughout the whole mid-Atlantic pipe. So for example, I'm gonna go over here to what they call their Long Island region, this yellow. And these arrows represent the paths that the larvae are taking. So you can see larvae that come from Long Island, some come back to Long Island, but also some get spit out into New Jersey. But the point here is there's kind of this downstream flow of larvae for surf clams and shelf waters. But we don't know what the small scale patterns are. Again, going back to my recruitment model, I found that these three different longitudinal regions on the south shore of Long Island, they each experience different interannual patterns in recruitment. And there are obviously many reasons why they could be experiencing different recruitment patterns. But I suspect that dispersal, or rather settlement, or more specifically settlement success, is a big part of why those recruitment patterns different in those three regions from year to year. And again, settlement success is something we can quantify from these biophysical models. We also have an idea of what the dispersal distances are. However, that idea is very broad. So this study found that the mean dispersal distance for surf clam larvae in the Mid-Atlantic bight is 119 kilometers, but with a standard deviation of 94 kilometers. Now, that's a huge spread. And there are a few reasons why that spread is so big. I believe one of the reasons is that they didn't empirically measure their behavior metrics. You know, they tried to model swimming speed, model sinking speed, as opposed to actually using experiments to get those values. And I suspect if we fine tune the behavior, we can either restrict this range, these dispersal distances, to have a better idea of what they are, or we can figure out, you know, what behavior patterns lead to what specific dispersal distances. And I also believe that climate change will affect, or may affect, you know, these behavioral data, all these biological inputs, which may then change the dispersal distance. And we also have an idea of what the settlement success rate is in different regions. So this figure, it's not the most intuitive. Let's just look at this row right here. So LI, again, Long Island, New York bight. These numbers represent the percent of larvae that are reaching this area, not necessarily settling, but reaching. And we can see that in Long Island, most of the larvae that are reaching Long Island actually come from SNE, Southern New England. But Long Island also supplies a bunch of its own larvae to itself. Again, this is just the area that the larvae line up. If we look at what the settlement success rate is, these numbers change drastically. We saw that Southern New England supplied most of the larvae to Long Island. But most of those larvae that come to Long Island waters from Southern New England are not successful settlers. Most of the successful settlers in Long Island come from Long Island. But what's funny is most of the larvae that Long Island produce don't wind up in Long Island. They get swept downstream to New Jersey. So Long Island's kind of getting the short end here. You know, it's making larvae from New Jersey. It's not getting larvae from anyone else. And again, I suspect that these settlement success rates will change if climate change is affecting all those behavioral metrics and those biological inputs that I was talking about earlier. Which brings me to my hypothesis, which you probably can put together by now. And so I suspect, again, climate change will affect surf clam larvae behavior, which will change larvae dispersal patterns. And again, behavior, we talked about this already, swimming speed, sinking speed, larval duration. And by dispersal patterns, again, I mean dispersal distances, settlement successes in different regions. Again, I'm going to look more in New York waters, eastern versus western waters, onshore versus offshore, those types of things. And so how do we get the behavior data? Well, that's where DEI comes in. So thanks to Rob and his awesome OA lab, we can manipulate OA scenarios and temperature to measure all of those behavioral metrics of interest under these different climate change scenarios. And so this represents our experimental design. We have two pH treatments, 7.7, 7.3, and we have 3 temperature treatments, 23 degrees Celsius, 20 Celsius, and 17 Celsius. And we have 5 replicate tanks per each treatment, fully factorial experiment, of course, which leaves us with 30 total tanks that we have to maintain. And so you're probably saying, well, Ray, how the heck did you choose these values? So 20 degrees Celsius represents present mid-water temperatures in inner shelf Long Island waters. So again, middle of the water column here. 17 degrees Celsius represents time-casted mid-water temperatures back in 1970. And 23 degrees Celsius represents projected increases in mid-seawater temperature to about 2100. And our pH value, so 7.7 is within the range of possibilities of inner shelf waters off the South Shore of Long Island. And 7.3 represents a 0.4 drop in pH, which is potentially forecasted for this area of the world by 2100. And so the idea here is if we compare this scenario to this scenario, we may be able to provide a mechanism or an explanation for some of those recruitment patterns that I observed. And if we compare this scenario to this scenario, we may be able to predict what's going to happen to larvae in the future. And kind of nested within this is a half-ish Gulf of Maine application. You know, you could say that 17 degrees Celsius represents present temperatures, not around here, but in the southern Gulf of Maine, and 20 degrees Celsius may represent that near 3-degree increase in the Gulf of Maine that Rob was talking about by year 2100. So if we compare these two scenarios, we may have an idea of what's going to happen to larvae, circling larvae in the Gulf of Maine. And so how do we get the behavior data? Well, for swimming speed, it's pretty simple. You put the larvae under a microscope, you take a recording, and then you use ImageJ to literally quantify the swimming speed in pixels per second for each particle, or in this case, each larva. It's really not that complicated. It's an easy metric when it comes down to it. Now, time to settlement or pelagic larval duration. Currently working through this, and right now, the way I was trying to quantify this is I'm looking at the percent of larvae that have their foot out looking for a substrate. In other words, I put 50 larvae underneath the microscope, I count the amount that have their foot out looking for a substrate, I count the amount that don't, turn it into a percent, and voila, that's the percent of larvae that are ready to settle at that time point. And now sinking speed. Not too sure yet. This is something we can do on preserved larvae. So this is not something we're measuring right now. I could potentially model it from growth, but as I talked about earlier, I want empirical data, not model data. And if the larvae were big enough and I had good enough eyes, I could put a graduated cylinder in front of me with a stopwatch and watch them fall and time them that way. More fancy version of that is having a camera, that's kind of rigged up to some sort of water column structure and measuring it that way. I'm hoping I can get my hands on that type of rig as opposed to having to do it with my naked eyes. But we'll see what happens there. And because we have time and resources, we are also measuring a bunch of other things that may not go into the final physical model, but are still ecologically relevant or just interesting. That we're measuring absolute growth, mortality, mineralization, clearance rate, respiration rate, immune response, i.e. mortality after bacteria exposure. We got a whole bunch of things that we're measuring. And so where are we now? Well, we are on day 22 of our experiment, and as Rob pointed out, we are exhausted. In addition to husbandry and maintenance of the day, we've been measuring these particular things on these particular time points. You can see I've gotten 4 time points for swimming speed and fixing larvae for things we can measure on preserved larvae. Actually, I meant to put an X in here because today, just before I came in here, I was doing some swimming speed trials. Clearance rate measured on these 3 days. Respiration rate, we finished up the first set of trials a few days ago, going to start again tomorrow. The bacterial challenge, i.e. the immune response, has certainly been a challenge. You can see it's one of the most time-consuming responses here. We have 2 trials there. And then PLD, pelagic larval duration, we started that on July 5th, because on July 4th, when we were sieving and cleaning, we noticed a bunch of larvae were kind of sticking to the sieves. And we suspected that may be because they're ready to settle. Sure enough, we look at the scope and we see plenty of foot activity. I also want to take this opportunity to say, we're measuring a lot of stuff. And the reason we/I have the ability to measure all these things is one, because of Rob's awesome OA lab, but two, because we have an intern and an assistant, Cassie and Hannah here, who have really just helped expedite things and have allowed me to measure all these cool things that I'm interested in. So I just wanted to take a moment to thank them. Even though nothing's done yet, we don't have any data, all the data may be useless, but we're at least trying. I'm able to try so much with this. And with that, I will take questions.
You did a really great job explaining everything. Just saying.
Are there any questions for Ray?
Parker, get set.
Thanks, Ray. Really exciting to kind of unpack some of these dimensions around dispersal and settlement. And at one point, kind of the crux of one of the management implications that you spoke about is this idea of, rotating the crop or rotating the harvest area as, one fundamental option for getting better yields per area and protecting some juveniles. So with your insight on the effects of climate change on settlement and recruitment, how do you think this kind of data might ultimately influence what is ultimately a social question and a management question? It's not just one garden out there that we're thinking of rotating the crop. It's in many ways a tragedy of the commons scenario where everyone is vying for a similar area. So how do you think this kind of information could influence the future cooperatives around shellfish management?
So I will do my best to answer that question, but I am new to the management world, and so I'm still learning some of the terms that you mentioned. And I don't know if I'm going to actually answer your question, but again, my idea is, we can have policymakers tell fishermen, hey, during this year, leave this spot alone, and then the next year say, okay, go for it. I don't think that answered your question, but that's the best I got for you as of right now.
Matthew Lehman asks, how do we know that the surf clam is not overfished?
There are state and federal surveys and studies that have loads of data and have particular analyses to see if the amount of individuals that are being removed is past a certain threshold. Now, I don't know the ins and outs of those analyses off the top of my head, but that's the idea. And we're well below that threshold for surf clams. And just as an anecdotal piece of evidence, at least in New York, I know of only 2 surf clam fishermen all throughout the South Shore of Long Island. That's over 100 miles of coastline, and there are only 2 fishermen left. So, that speaks at least to the status of the fishery and whether or not overfishing is prevalent in that area.
Bill Mook.
Hi, thank you. That was really interesting. And I guess this is probably more applicable to more estuarine species, but our observations over a few years has been that these larvae are really sensitive to climatic events. And I'm wondering, in other words, it's unusual for the pH to be held at a given value or a saturation state at a given value for the duration of the larval phase. So I guess my question is, how do you factor that into the models? And it could be a lot of different environmental variables that change with big climatic events, whether it's large runoff events, big wind events that would affect dispersal. And I guess, how do you even think about that when you're trying to figure all this stuff out?
Yeah, so, I talked about trying to model dispersal patterns in different scenarios. And essentially, the scenarios are going to be extremely limited. We could try to interpolate what's going to happen between the scenarios that we're testing out. But, I'm not a big fan of interpolation. And so ultimately, we really will only be able to say, under this specific scenario, this is what happens. And so to your point, the applicability of that, when you consider how much, pH temperature changes on a, quite frankly, daily basis, the models will be limited. One way to maybe fix that is, in the future, we could measure these responses under more than 2, pH levels or more than 3 temperatures and try to establish a relationship there. And then that will help covering, these in-between scenarios that are not really targeted by our intentions right now.
Is it, I guess, a follow-up question, is it too complicated, difficult, or expensive to do more actual in situ observations? So, and I'm thinking back to a few years ago when we were looking at, we were actually looking at mussel larval development in the Downeast River because it's a fouling organism on our gear. And one of the things we observed was that there seemed to be sort of a protracted spawning period, and that we were able to see larvae in the water, but in that particular season when we did not see a set of mussels, we did not see the larval development occur. So we were seeing a lot of early stage larvae, but they never progressed, which indicated to me that there was some environmental condition that was preventing that. And I guess that's kind of maybe an extreme case, but because obviously surf clams are a more oceanic species, the environmental conditions are generally more stable. But I guess it gets at a little bit of what I'm kind of concerned about is how we evaluate what is it that's causing recruitment failures.
Yeah, so Tom, I think you hit on something important when you first said, you're familiar with estuarine species. It's very difficult to get in situ data on shell species. It's just extremely expensive to spend that much time on the water and just, you're hoping that you catch, larvae. That being said, you can't rely on those studies as the main, those types of in situ observations as the main focal point or as something that you can draw a conclusion from. But with even some patchiness in those in situ observations, what you can use those to validate the model. And that's something that if I have time, I would love to do during my dissertation. So I agree, in situ data is needed. It can't be used as the foundation for shell species, but can definitely be incorporated to improve the model and validate.
Thanks.
Any other questions?
Yeah, George asks, how is time at pH or temperature considered? The duration of stress or the relationship to growth stage?
Can you bring that up again? Yeah, breathe in and think about it for a moment. Not too sure I understand the second part of your question, but we're keeping the pH and temperature constant for these 3-4 weeks. So the duration of the stress is essentially the whole lifespan of the larvae, or at least our scenarios here. But again, going back to some of the other questions that were asked or brought up, it's not necessarily the most environmentally relevant. And so, kind of mixing and matching different pHs, temperatures for different times would perhaps be informative for the future.
I've got a question, Ray.
Yeah.
Or maybe for Rob. Is it possible to program in some kind of variation, either in temperature or OA across time. I mean, we're talking about static treatments for the duration. Maybe that's what he's talking about
Yeah,
here.
yeah.
Is there a way to manipulate, and that might get at Bill Root's
Yeah.
question as well.
Yeah, I'll hop in here. So we can absolutely do that in our lab. We're using the Neptune Apex system, which is it's an aquarium controller that was designed for like reef aquarium hobbyists. And it has the ability to ramp our experimental set points up and down over time to achieve a sort of diurnal variability in pH and temp and other things. And that's something that we're hoping to leverage in the near future to hammer out some experiments like that.
Dustin adds in the chat, an easy way to do that is using a flow through that maintains a delta between treatment and controls.
I had a question, right? Because I was thinking about all the different things that you're measuring. And I was thinking about some things that we learned with the soft shell clams. And I don't know if it might apply to like figuring out why we are having recruitment failures with the Atlantic surf clams. Is what we're finding is the warming water is causing increased predation once the clams have settled and recruited. And so that's another, effect of climate change, but it's not exactly the, effect of too warm water. The larvae aren't surviving. Once they settle, aren't surviving. And you've got a lot of things going on in your model already, but maybe that would be something else to.
Yeah, I mean, I suspect predation is changing through time with climate change, and I would love to investigate those questions with surf clams. But taking the approach of these predator exclusion field studies and doing that in 50 meters of water in the open ocean, it's very difficult. You can try lab studies or even in different habitats that are more manageable, but then the applicability of those types of studies is limited at best.
Right. And Parker, I guess that has increased.
Thanks. So a question for both Ray and Rob and maybe others from the Downeast Institute. Just having this new laboratory available is an amazing increase in the research capacity for the whole region. And, I imagine just totally celebrated. Have you had new opportunities for collaboration among others? Have other researchers been reaching out to you? And what do you see as kind of the position of your unique, laboratory capabilities for regional research in your area?
Yeah, so we've had a little bit of interest from researchers from other institutions and states. We had a few leads before the pandemic started, and then that happened and everything kind of fell off the table. Ray is our first collaboration in the new OA lab, which has been working out fantastically. And as for the capabilities of what we have built, we really made a point of emphasizing a high degree of replication. And so we can pull off these factorial experiments with multiple stressors to a higher degree of replication than a lot of the labs out there are capable of. And so I think we're in a really good position to collaborate with more research teams and get some good research out. And by the way, for anyone listening, we are actively seeking collaborations. If you have a project idea that you think we can help with, give me a call.
Is there any final thoughts, Rob or Ray, you'd like to leave people with?
Thanks for the feedback. You guys are giving me a lot to think about in ways I can improve some of my plans. So appreciate that.
Awesome. Awesome. Well, thank you, and thank everyone for coming. I hope this was informative, and we did record it, so we'll have that resource available for people if they want to review it or send it to one of their colleagues. We'll have that as well. So thank you everyone for coming to our first ever seminar about climate change.